Welcome to our presentation on roving Mars and the Mars 2020 rover, also known as Perseverance. As some of you may know, the rover's launch day is today, which conveniently coincides with our presentation time. Anyways, my name is Ethan Lai.
My name is Anushka Narayan.
I'm Swetha Palapur.
I'm Hannah Clay.
I'm Amelia Craig.
I'm Rachel White.
I'm Taran Lonavat.
I'm Gwyneth Wong. First, we'll be discussing what Jezero Crater is and why it was selected to be our landing site for the Mars 2020 rover mission. Jezero Crater was chosen as the landing site for the Mars 2020 mission for its high geologic diversity and because it contains several geological features that have a high biosignature preservation potential.
As the figure on the right shows, Jezero Crater was formed during the transition from the Noachian to the Hesperian era, which is when Mars's climate changed drastically. Jezero has a record of this change, and by exploring it, we can determine why it happened.
The Mars 2020 mission strives to determine whether life existed on Mars, to characterize the climate on Mars, to characterize the geology of Mars, and to prepare for human exploration.
We know Jezero Crater was once an aqueous environment due to the presence of minerals which are only formed in water. Now, let's explore 4 regions of interest, as seen in the figure, that fulfill the 4 objectives of this mission and observe the evidence of aqueous alteration.
One such region of interest is the Delta deposit. This region contains an abundance of sedimentary iron-magnesium smectites, which form in the presence of water. We can examine these smectites for organics and study their aqueous alteration to give us more insight into the early Martian climate and the hydrothermal environment in which the smectites were formed. Deltas on Earth are well understood, and the unique ability of a delta to capture the mineralogical blueprint of its entire watershed promises exciting possibilities for geological and astrobiological research.
As seen from this diagram, Jezero is one of the few sites on Mars where a carbon-based sedimentary rock called carbonate forms, and that too, a lot of it. Since Mars's atmosphere is carbon dioxide rich, we expected a lot of carbonates on the surface of Mars, but that was the opposite of what we found. This is the paradox of Jezero. Why are there so many carbonates at this location? Since carbonate formation in Jezero is not well understood, studying the formation pathway can confirm or disprove some of the hypotheses we have on how these carbonates formed. According to observations from satellites, the mineral olivine is closely associated with carbonates. But getting a close-up look with Perseverance can help us find any other minerals that could have played a role in carbonate formation. Studying carbonates can also tell us the conditions they formed in, giving us information on the early Martian environment, the atmosphere, and habitability. Here on Earth, carbonates preserved fossilized stromatolites, which provided evidence of ancient terrestrial life. A promisingly similar group, known as marginal carbonates, found along the rim of Jezero Crater, are also excellent at preserving possible biosignatures and perhaps past Martian life. So, the carbonate fill of Jezero is one of the most important regions of exploration for the Mars 2020 mission.
The Isidis region of Mars, near Jezero Crater, is thought to have been volcanically active. Studying this region is vital to understanding the timing of igneous events on Mars and will allow us to develop a more accurate Martian chronology.
As seen in this figure, this last region of interest has an abundance of clays or smectites that were recently exposed. Organic material is known to be highly concentrated in these offshore deltaic environments and can easily bind to these smectite clays. So the western deltaic deposits are a promising location to look for preserved biosignatures. In addition, studying the deposition in this area can tell us about the relationship between surface runoff and aqueous alteration.
Now we will cover the rover's instruments. What tools do we have aboard the Mars 2020 rover?
MassChem-Z takes colored images to characterize the Martian geology relating to the 3rd mission objective. MassChem-Z is located on the head of the rover's mast. In the top right, you can see an image of MassChem-Z.
MEDA measures the weather and monitors dust with sensors of the Martian surface. It is located on the rover's mast, deck, front, and interior of the rover's body, and its connection to the mission is with Goal 2, which is studying the climate on Mars.
PIXL is an X-ray spectrometer that measures the chemical makeup of rocks relating to the life and geology mission objectives. It is located on the turret at the end of the robotic arm. In the top right corner, you can see an image of PIXL.
RIMFAX analyzes geologic features under the Martian surface with ground-penetrating radar. You can see this happening in the top right image. It is located on the radar antenna, which is on the lower rear of the rover. It is connected to the mission with Goal 3, which is studying the geology of the Martian surface.
Sherlock searches for minerals, organic molecules, and potential biosignatures relating to the life and geology mission objectives. Sherlock is located at the end of the robotic arm. You can see an image of Sherlock in the top right corner. Watson takes images to assist Sherlock in its search for biosignatures. It is located on the body of the rover. The image in the top right was taken by Watson and displays the different minerals found on a rock's surface.
SuperCam investigates the chemistry, mineralogy, atomic, and molecular composition of the Martian surface. It is located on the head of the rover's mast, and it connects to the missions with Goals 1 and 3, which is studying life and geology on Mars.
MOXIE is a revolutionary instrument that produces oxygen from the carbon dioxide in the Martian atmosphere. This instrument tests technology that may be used in future manned missions to Mars.
And finally, we have Ingenuity, which is the helicopter on the Perseverance rover. It is 31 inches tall and it only weighs 4 pounds. Its blade speed is at 2,400 RPM in comparison to a normal helicopter speed, which is at 500 RPM. Its mission is to demonstrate the feasibility of drone flight on Mars, which is the first flight on another planet. If and when Ingenuity works, it will take images of untraversable locations.
Now we are going to talk about the mission control of the rover and how the rover traverse is planned. Here are 3 maps showing the engineering constraints of Jezero crater. The first is of rock coverage, the second is of the slope, and the third is of the types of terrain in the crater. On the rock coverage map, for example, the yellow and red areas convey the presence of larger rocks that the rover should avoid on its traverse, whereas the green means less rocks and it's okay for the rover to go over.
Perseverance moves through a combination of autonomous AI navigation and user control. Speed depends on the ease of navigating terrain, as shown by the left image. On easier terrains, Perseverance can process images of the terrain around it in real time and decide where to go, as shown on the image on the right. On harder terrains, the rover must go very slow as it is being controlled by someone and cannot process and decide where to go by itself. On our route, we want to find the easiest path to all our locations so we can be more efficient. Another factor in Perseverance's route traverse is regions of scientific interest and where we can collect and store samples. On Earth, there are many different strategies for this, such as traveling upstream and collecting samples source to sink, exploring minerals or deltaic deposits. In Jezero, we can explore the sink of the Jezero watershed, which is important because, as shown by the map on the left, the watershed has a lot of different terrains, each represented in a different color. Sediment from all these terrains collect in the Jezero crater delta, and Perseverance can sample sediments from all of these different terrains. Ideally, we want a route traverse that keeps the rover safe while also taking advantage of all the interesting science opportunities that Jezero crater has to offer.
Here are 2 rover traverse maps that we came up with in a lab that we did. You can see they are slightly different. This is because there is no perfect traverse. We weighed the scientific value of certain areas compared to the engineering constraints and planned out a route from a landing site to an objective site. These are the same constraints that the scientists will use to plan the final traverse route that Perseverance will take.
After deciding on the location, we plan out what to do at each location. For example, on the left is a sample daily schedule from Curiosity, with time at the top progressing from left to right and instruments on the left. Blocks on the schedule represent when instruments will be operating. Planning out instrument schedules can be challenging due to the constraints on the instruments and the time each instrument takes to operate. For example, some instruments cannot operate together, and some of the more interesting regions are too far away for the arm to reach, so we are limited to only using cameras to study those regions. We plan out the instrument schedule to make sure not to overexert battery resources or compromise data.
We will now move on to the topic of astrobiology and the search for life on Mars. Perhaps one of the most important aspects of the Mars 2020 mission is to search the planet for potential biosignatures. A biosignature is a substance that indicates whether life existed or still exists in a location.
There are several categories of biosignatures, the first being molecular, which includes things like biomarkers. A biomarker is a substance whose presence in a living system is indicative of a phenomenon. For example, lipids and structural biopolymers are classes of compounds known for their stability under harsh environmental conditions. Another example is that lower temperatures on Mars would aid in the preservation of hydrocarbons. There are also isotopic biosignatures, fossilized biosignatures, and morphological biosignatures, which includes things like microbial mats. Biofilm forms when bacteria adhere to surfaces in moist environments by secreting a glue-like substance. On the right, you can see an image of a microbial mat.
Biosignatures would help answer many questions scientists have about potential life on Mars. Currently, scientists have developed multiple theories about life forms on Mars. One theory is that Mars once contained anaerobic chemotrophs. Anaerobic chemotrophs are organisms that obtain energy through chemical processes, especially oxidation. Another prediction is that life on Mars consists of microorganisms and bacteria. This theory has been developed from scientific studies done in extreme regions of the Earth. For example, in a study done in 2003, a NASA scientist discovered a new microorganism that thrived in a low-oxygen, salty lake within an enclosed volcanic basin on Earth, which is similar to environments on Mars. These similarities between environments may indicate that Martian life may likely consist of extremophiles, or microorganisms that can survive in extremely harsh environments. A third theory is that life formed early on in Mars's history, before or during the Noachian period, a period in which water is believed to have been present on Mars.
In the Syrtis Major Quadrangle, there are several regions that have a high chance of harboring biosignatures. One of them is the Jezero Crater Paleolake Basin, which may contain high levels of detrital phyllosilicates. These are crystalline calcium carbonates that are strongly associated with the fluvial activity that took place in the Paleolake Basin, which is surrounded by the western and northern fan watersheds, which you can see a picture of on the bottom right. It has been widely predicted that these terrestrial lake sediments were transported into the basin from the surrounding western and northern watersheds, as opposed to having formed in situ. If this is confirmed, these sediments can reveal information about rock weathering, water on Mars, and potentially life. In addition, there is evidence for fluvial crossbedding in the western alluvial fan, which you can see a picture of on the top right. The direction the crossbeds are dipping indicates paleocurrent, the rough direction of sediment transport, which can ultimately tell scientists about weathering and how the fluvial environment was like. As you can see, the common theme behind all of these deposits is that they're all associated with water. These deposits most likely have high biosignature preservation potentials and can tell us a lot about life and chemistry on Mars. This is due to the fact that abiotic organic molecules could have dissolved in lakes and seawater.
Hydrothermal minerals found on the Syrtis terrain are also a sample type of interest because fossil remains of chemotrophs are sparsely populated except in the vicinity of hydrothermal activity. A possible explanation for this concentration of fossils is that hydrothermal vents contain a large amount of available nutrients and thus could sustain life.
Shown here on the right is a picture of a microbial population that was discovered in Antarctic permafrost in 2007. Even though microbial viability can be sustained with extremely low amounts of energy, as shown in this photo, scientists doubt the same will happen on Mars because of the crustal radiation, limited availability of fixed nitrogen, and nutrient-poor Martian subsurface. These conditions make carrying out basic cell maintenance tasks like DNA repair, protein renewals, and cell division challenging. As for the conditions that make life suitable on Mars, Martian anaerobic life could have undergone metabolism using a number of electron sources present on Mars, like the hydrogen gas from hydrothermal activity. In addition, anaerobic and aerobic life may have used organic matter of abiotic origin to undergo metabolism.
Implications. What does Mars 2020 mean for us on Earth? And what does it mean for a future manned mission to Mars? Aboard the Mars 2020 rover are 5 spacesuit material samples. Analyzing the effects of the Martian environment on potential spacesuit materials will help engineers keep our astronauts safe. The picture shows the 5 materials all hooked up to Sherlock, the instruments which host the materials. MOXIE is an instrument that converts carbon dioxide to oxygen. Aside from providing oxygen to the astronauts exploring Mars, the success of MOXIE provides crucial benefits for the return mission back to Earth. Launching a manned mission requires hundreds of thousands of gallons of oxygen. It's highly inefficient to lug all that oxygen to Mars, only to use it to return back. So what if we could make that oxygen on Mars? That's the value of MOXIE. MOXIE will allow us to create oxygen that can be used to provide breathable air to astronauts, as well as to power the return trip back to Earth. And lastly, as Gwyneth and Ethan mentioned, there's a decent chance we'll find evidence of previous life on Mars with the data collected from the Perseverance rover. Finding life on Mars would be the first sign of life on another planet and the oldest life ever discovered in the universe. Life found on Mars. How's that for a scientific breakthrough?
Another exciting prospect for this mission is getting actual samples from Mars back to Earth. The Mars 2020 mission is the first mission to be equipped for a sample return mission. Perseverance can now collect samples which can be retrieved through later missions. On returned samples, we can search for chemical and isotopic evidence for biosignatures. We can even analyze carbonate samples brought back from Mars to find out the temperature, pH, and salinity of the water they were formed in. Returned volcanic samples can be radioactively dated to determine exactly when significant geological events took place on Mars, which can give us more accurate timescales compared to the current method of crater counting. With Mars sample return, advanced testing that wouldn't be possible on-site could be carried out in labs on Earth, further leading us to enticing and exciting discoveries.
Thank you, C's. Here are some personal statements on how this internship has impacted us. Personally, I really enjoyed getting to hear about the lives and work of scientists and engineers through the C's speaker series.
I really enjoyed collaborating with my teammates and gaining insight from my mentors while working on the challenging lab activities.
I loved learning all about the Perseverance rover and the Mars 2020 mission.
I enjoyed meeting and working with people with the same interests as me.
I loved understanding more about what people do in the field and at NASA.
I really appreciated the opportunity to talk to and learn from scientists who work in the fields I'm interested in.
I loved learning about Martian geology from my mentors and my science role models.
These gave me the confidence to unmute on Zoom and ask a question. Some of the most memorable aspects of our internship actually came in the form of setbacks. A few of the difficulties we encountered including trying to balance scientific and engineering constraints. We wanted to maximize the potential for discovery while making sure we kept our billion-dollar rovers safe. It was also challenging to divide our work, yet still present cohesive labs. But this is a great team, and we worked through it together.
Now for our acknowledgements. We'd like to thank our wonderful mentors, Mike and Valerie, for everything that they've done for us, and to thank everyone who has organized this event. Here's a short list of our references, and does anyone have any questions for us?